We got some gentlemen here from UC Berkeley who I think will give a fantastic presentation with neural decoding. But actually, I wanted to have a student introduce them. This is Nick DeSisto. He's actually a computer science student. He works with Thomas at MUSC in his lab. Actually, Thomas came from this group at UC Berkeley, and Nick is doing great work out there. I'm super excited. Actually, there's no physics people in here, right? We kind of like hijacked a physics student from into computer science. But anyway, I'm going to hand it over to Nick, and he's going to introduce them, and then Nick is going to introduce the guys from Berkeley. So hopefully enjoy, and I'm sure it'll be super interesting. All right, thanks.
Hey everybody, thanks for coming out. We have today with us Alexander Huth, and we have James Gao, and they're going to talk about their research in neural decoding. And big thanks to these ladies. Without them, we could not have pulled this off. And also, here's a list of some of their accomplishments. As you can see, the research has been featured in Time magazine, Nature, Popular Science, as well as on PBS and Through the Wormhole with Morgan Freeman. So there's a lot of cool stuff there. So without further ado, James is going to take it away.
All right. Thank you. Thanks for hosting me. And I just want to talk to you guys about some of the, some of the concerns we have about looking at brain data as collected by MRI. So in my lab here, let's just open this up. In my lab here, we actually collect a lot of data using a technology called MRI, magnetic resonance imaging, and specifically we use functional magnetic resonance imaging. This means we collect multiple images through time, which allows us to see what's going on inside the brain. And this is a viewer that I developed to allow us to actually look at this data, because although this data is extremely high, high density, there's a lot of information in there, it's actually very hard to look at it. And this is actually a little short demo I programmed up for a museum exhibit that was on display at the Exploratorium for a while. So it's a little interactive thing. You can actually kind of wave your hands around to get it to do interesting things and look at some of our data here. So what you're seeing on the left here, let's scrunch this back up here. What you're seeing on the left is someone's brain activity played in real time while they were watching this video here on the right. Ooh, forgot my laser pointer. Hold on one second. All right. So this is someone's brain activity plotted in a red-blue color map. So blue means lower activity in the brain, and red means higher activity as they're watching this video. So this is what, now that we have this correspondence between what they saw and what their brain did, we can start developing models that map between the two. And using one of these such mappings, we can actually create a guess for what we think they saw inside the magnet. So this is what they actually saw, and this is what we think they saw using only their brain activity. So you can see that there are certain details. So for example, faces work really well in this case because due to details on how we actually did the decoding. And Alex will probably touch on some of this stuff later on. But I'm going to mostly talk more about how we actually set this viewer up and some of the considerations when plotting MRI data. Oops, other way. No. So one of the key things to know about looking at MRI data is that MRI actually gives you a volumetric view of the brain. This means that there's actually a recording, some information about tissue properties throughout a 3D space, one point in every 3D space, and it gives you this dense grid of information. This means there's actually a lot of information that you can actually use to model brain activity. But unfortunately, when you're trying to look at MRI data, it's very hard to do. That's because of the innate 3D nature of the data makes it such that, you know, things in front occlude what's behind. And even with this really fancy view that took forever to render on a normal desktop computer, it's very hard to understand what's going on. So what you're seeing here in these green dots is actually a volume integration view of the density of the tissue And I've actually already filtered this for intensities that should contain brain. So you can see kind of inside here, there is kind of the shadow of a brain, but you'll see that even though I filtered specifically for brain tissue, there's still a lot of like skull and things out in front that aren't really relevant for us. So there's a nice benefit to looking at MRI data, and again, it is that you have a lot of it. So if we scroll through that volumetric data now, slice by slice, you can see a lot of tissue properties kind of propagating through. And you can see light parts that have a certain magnetic property, darker parts have a slightly different magnetic property in the tissue. And as we pause on one of these slices, you can see that there's this characteristic kind of shape and color. And if you blow it up here, you can see that there's this lighter part here in the brain. This is called the white matter. And then outside along this like edge right here, it's almost like a rind on the brain, and that is the gray matter. Now, the key thing with this is when you're talking about human behaviors, the majority of interesting human behaviors actually are manifest inside the gray matter. You can think of the white matter more as like a wiring bundle that connects pieces of gray matter together. So the nice benefit of looking at MRI data in the way that I'm going to propose in just a minute is that you have basically a very small amount of it that is actually doing interesting things. And everything else you can try to ignore because there's very little, the way that we're imaging it means that there's very little variability there and there's nothing that we can actually model. So now that we can establish that there's this kind of thin shell around the outside of the brain that actually has the interesting information, what can we do to actually visualize it? Well, we can actually create a model of that shell. And we can do that by using a lot of computer vision algorithms. So in this case, I've actually taken one of these slices. I've used a special computer vision algorithm that knows things about the brain and segments out the surfaces. And you can see there's a yellow line that outlines the white matter, and then this red line that outlines the gray matter. So once we have this kind of a segmentation of tissues in the brain, we can create a voxelization mask of the data. And using this mask, you can actually slice it out into 3D, which gives you kind of a rough surface representation of the brain. And you know, this representation is not particularly useful because it's all blocky and from the voxelization. So this was actually just generated with a very simple Marching Cubes algorithm. We can apply a lot of information about how the brain actually folds and generate a surface, a very high fidelity surface that looks like this. So although this process, I made it sound really, really easy, this is actually a very intensive purpose. And unfortunately, there's a lot of topology fixing that has to go on between getting one of these masks and actually creating a surface like this. But the really cool thing is, now that we have one of these surfaces, you actually have a lot of things that you can do with it. So one of the example things is that you can manipulate it using geometrical operations. So for example, in this case, I took one hemisphere of the brain, one half of it, and we can blow it up like a balloon. This lets us see inside the folds of the brain. We can also cut out small pieces of it in areas that we know don't have interesting activity and flatten it out onto a sheet. This way, it lets us actually see all of the interesting portions of the brain all at the same time on a 2D figure, which means you can actually take one of these cortical flat maps and actually print it on a paper, and it lets you see all the interesting activity that occurs on it. Now, Alex is actually going to use a lot of these flat maps in a little bit because this is a great way to look at the whole brain all at the same time. But one thing you have to keep in mind is that because we applied, this is a very complicated folded 3D surface. So there's a lot of distortion that has to occur that is unavoidable when you flatten the surface this way. So this is a map of the distortion. Blue means that that area has been kind of shoved together and contracted, and red means it's been kind of stretched apart and expanded. So when you're interpreting anatomical areas of the brain, like what is where in relation to other things in the brain, you have to keep this map in mind to make sure that you're not tricked by how much of the cortex is actually devoted to any certain area, because there is a distortion map. So now you think that we have this really high fidelity surface, we can start plotting brain data to it, right? Not quite yet. So in MRI, there's actually a very strong trade-off between spatial resolution and temporal resolution. So we generated this surface using a very high-resolution anatomical scan. One of these scans takes almost 5 minutes to collect, even with the latest generation acceleration techniques. This is what allows us to actually see all these really detailed curves and folds inside the brain and actually lets us segment them. Unfortunately, if you take a snapshot of the brain once every 5 minutes, you've basically mushed together all the things that happen in the brain over a 5-minute era, and you can't really do any interesting modeling in that case. So here's a different kind of image. This one only took 2 seconds to create. We took a snapshot. We can actually collect one of these images once every 2 seconds. And in fact, this has been optimized. This image has been optimized to give you a maximum contrast between a measure that we can actually use to model the brain. Specifically, using different shifts in magnetic fields, we can actually see basically oxygen usage within the brain. The oxygen usage is basically a proxy for what's actually happening at the neuronal level. It's averaged over a pretty large area in this case. This particular scan is about 2.5 millimeters on a side, and with about 4 millimeters in the slice. So each one of these pixels, basically, in 3D, we call that a voxel. Each one of these voxels is about 2.5 by 2.5 by 4 millimeters. So even though the resolution is relatively low compared to this image, we can actually see it fast enough to actually give us interesting information about the temporal dynamics of the data. So now we have this problem where we have a really high resolution surface, and then we have a totally different resolution image. We're going to have to match them up so that we actually know how do we project the data from here onto the surface. Well, there's a nice, easy way to do that, and that's through just a very simple affine transform. Since we have a bunch of vertices that define this surface is a triangular mesh surface, we can pass all these vertices through just an affine transformation and actually find their corresponding location inside the image. And if we do that for one particular slice, you can actually see we can get pretty good alignment. This type of alignment is constrained by a lot of the physics in actually acquiring the data. So even though the anatomy never changes, so like the brain's not going to sprout a new fold when you're 40. That doesn't really happen. So you can be sure that at least the small scale structure of the brain is going to be the same. However, the key is due to how quickly we collect this image, there's actually going to be a certain amount of stretching and kind of shifting around in the data just due to how we actually collect the data. And you basically try your best to make your affine transform work, but it's not always the best. There are some nonlinear methods to do this, but nonlinear co-registration is a big can of worms. So we typically just use a very simple 12-parameter affine transform, and it works pretty well most of the time. So now you're thinking, well, we have the data that's been matched up. Each vertex has been plotted into the space. We can do something very simple to actually get all the data onto the surface and allow us to see it all at the same time. If you're familiar with any computer graphics, you know that a standard vertex color pipeline is probably something that you can use to do this kind of plotting. So how a vertex projection typically works is, let's say this is a small piece of our surface embedded in our 3D grid of data. Okay? All we would do for a vertex color pipeline is we figure out where the vertices are, we project them into the volume space of the data, and we basically just sample the data at these vertices. We collect what color they should be in the final data, and then we take our surface, we push it through our standard frustum projection. So we plot it now on a 2D screen. So now this grid is the pixels on your computer screen. Now, standard with any vertex color pipeline, you basically rasterize this data and you get an image that kind of looks like this. This is not quite right, though. Like, we know that there's a red voxel here. We know that there's a blue voxel here. We know there's a green voxel here. But there's no weird grayish mixture color voxel in between. So even though this seems like a great idea for vertex colors, it's not right when you're thinking about the actual data. And unfortunately, with a fixed function OpenGL pipeline, this means that you can't really actually do the correct thing, which I'm going to tell you in just a moment. With the use of fragment shaders, though, and a programmable shader pipeline, you can actually do a lot more interesting things. So what you can do in the pixel projection, which is a thing that we implemented in pyCortex, is you can actually use each individual pixel to sample the original volume space. So most of this pipeline actually progresses about the same way. You take all your triangular mesh surfaces, you push them through your Frustum projection transform, so you run them through your vertex shader, and you get them into the screen space. So now you have your fragment shader that's running through each pixel at a time. But instead of actually just linearly interpolating between the colors at the vertices, what you can do now is actually take the pixel and directly project it back into the volume space to look up the color that it should be. So let's say we're trying to figure out what this particular pixel should be. We take this pixel, and when the fragment shader hits it, it's going to run a small bit of code that says, project this pixel back into the volume space. What should it be? So we take this pixel, we look up its value inside the volume. We see that it's supposed to be a red pixel, so we assign it the red color. Let's do it with another pixel. In this case, we look at its value back in the volume space, and we see it's supposed to be a blue pixel. You repeat this over and over again, and you actually get the original data back. This gives you a very high resolution reconstruction of what's going on underneath the data, and allows you to actually see hard edges in your voxels, which is previously unattainable using just a fixed function pipeline, or rather it would have been extremely expensive to render in real time. So another way to think about this is to look at this in 2D. So here's your eye, and here's a 1D screen. And these big colored boxes here are your volumetric data. And this black line here is the surface that's cutting through your data. These red X's are your vertices that sampled the data. So this vertex gets assigned to this light blue color, and this vertex gets assigned to this dark blue color. If we do our standard vertex shader pipeline, or just our standard fixed function pipeline, it's going to sample the data at these 2 points and just linearly interpolate between these two. So even though this surface actually cuts through this orange voxel, you never see any orange on the screen. And this is not very accurate. With the pixel, so when you actually render the image, you actually can't really see the edges of the voxels. You get these kind of streaky figures that may correspond to certain voxels, depends on the density of the vertex sampling. But you're never going to get the original voxels back. With pixel mapping, now every single individual pixel actually gets projected back into the volume space and lets you sample the data. So you can see that this surface now actually does capture the orange voxel. And if you actually look at how the surface gets rendered, you can actually see boxy things that look like they should be voxels. So another way to look at this is on the actual rendered thing. So now we've actually taken our surface, and now we've used it to sample data from the low resolution functional image. And you can actually see that there's colors that look like boxes in here. And here I've plotted in red a single example voxel that's an actual physical cube projected into the data. And zooming in, you can actually see that cube corresponds to your actual data. And since this is actually running through a fragment shader, it's really fast. It's highly interactive. You can see I was just playing with the data and kind of moving it around and all sorts of things. And all of this is in real time. And specifically, it's all accurate. So if you zoom in on this one patch of your data, you've actually devoted more pixels to sampling the data. You're actually able to see more of the data as well as you zoom in. So this is a very flexible, very adaptive way to look at your data. And as far as we know, no other graphics package actually samples volumetric data in this way. There's another cool thing that we can do that a lot of other packages when analyzing MRI data does not do. And this is something called thickness mapping. So up until this point, I've been kind of assuming that the surface that we generated before from the anatomical data, we thought that it was basically one infinitesimally thin sheet that's cutting through all your volume. But we don't have that. The cortex, that gray matter, has a thickness. And a lot of the times, a lot of these software packages don't actually think about the thickness of the cortex. So what we can do here, since we have both surfaces, both the inside and the outside sheet, is we can do something called thickness mapping. So here again is that 2D representation of our data. Here's the screen again. Here's the eye. Now we have two surfaces, the inside surface and the outside surface. Now when we project a pixel into the volume space, we can actually sample along a line that connects the inside and outside surfaces. We can collect multiple samples along this line, average them together to give you the actual data in there. And when we're implementing this in OpenGL, we don't actually have to make every sample. We can dither the sampling, subsample everything, and make an image that actually looks like this. So now you can still see that there's boxy, voxel-like things in your original data, but now you actually get gradations because it cuts through a thickness through the cortex. And this is a really cool way to look at the data that is, again, pretty unprecedented. There's one additional concern when you're talking about plotting the data, and that is how do you sample the data? So again, up until now, we've been kind of assuming that all of our data exists in these big boxy squares. And so here's our underlying functional data. Here's our surface that cuts through it. That's not quite right. We're using a physical process to actually measure what's going on inside the brain. So we're never going to get sharp edges like this. What we actually see probably resembles something closer to this. You get these little orbs with a lot of ringing artifacts because we're actually collecting this data using a Fourier space imaging technique. So if we were to directly sample this data, if we took this particular vertex and just said, it is blue, that's not quite right either. So another way to think about this is in terms of filtering, right? So this pixel is basically trying to filter this data. And if we do a box filter over the data, which is basically a nearest neighbor sampling, it's going to just say this whole box is just blue. We don't care what it is. Not quite right. Ideally, what we want to do is actually use a sync filter, because this gives you the optimal filter. A sync filter will actually downweight around the edges and upweight around the middle, and actually gives you the correct sampling given the constraints of the imaging technique. Unfortunately, this kind of filtering is very, very expensive. That's because you have to do multiple texture lookups all on the same call, and this is not very efficient in OpenGL. So we can't really quite do this in real time. But what OpenGL actually does let us do is use linear filtering. So we can actually, if this vertex samples right in between the two, we basically just linearly blend in between these two voxels. And we can do this in 3D to actually give you trilinear blending. This is not quite the most accurate thing you can do, but it is fast and it lets you actually see the data in real time. So if you're familiar with OpenGL, you know that this one's called GL nearest in the filtering language, and this one's called GL linear. So the sampling does make an effect on how you actually look at the data. So here we have a kind of a zoomed in view of nearest neighbor filtered data. And you can see, you still see those boxy squares that actually indicates sharp voxels. And while this is a great way to know that your original data consists of little boxes, it's not accurate. This is actually plotted using trilinear filtering, and you can see that it's much smoother and potentially easier to understand what's going on. So now that I've told you all about the concerns of how we actually plot MRI data onto a surface, I'm going to show you how we exactly did this in pyCortex. So pyCortex is an open source software that we wrote for plotting these surface visualization, and you can go find it on GitHub or just go to pycortex.org. And it's actually mostly, the user will mostly interact with it through Python, but as soon as you get all of your data in through Python, you can actually get access to it through a WebGL interface. So why did I program pyCortex in WebGL? Well, the number one key thing with scientific data visualization is that it's usually very hard to do. It's a real pain in the ass. You have to take, tons of software, install it, and then make sure that it all works. It's very hard to do. But with WebGL, you point your web browser at a website. If any of you have a computer there, you can actually just go to this website right now, and you'll see that it just works. You pull it up, and you don't have to install anything on your computer, and you're looking at data already. It's also cross-platform. You don't have to worry about a Mac install or PC install or even a Linux install. It just works right out of the box. And again, since this is actually in a web page, you can actually post your web page somewhere on the internet and point your colleagues, your collaborators, or even the general public at it. And it lets them see the impact of your research right up in front and in the most scientifically accurate way possible. Another thing that WebGL gives us is actually, since it is fundamentally through a web technology, you have access to a lot of JavaScript libraries that lets you do very interesting things with interactivity. So earlier in that demo, you can see that I've actually matched up in time, real-time stimulus versus the real-time response. And this is something no other software actually lets you see. No other software for doing visualization lets you look at the simultaneous use of your data. So WebGL has a lot of cool advantages that lets us do these kind of interesting things to look at our data. But it does have its limitations. So if any of you are familiar with OpenGL and how it's implemented, you'll know that WebGL is kind of a very small subset of OpenGL functionality. So for instance, it really only has the GLES 2.0 functionality. So you're only guaranteed that functionality. And one of the biggest, most annoying limitations is that you can only use 16-bit index arrays. That means if you have to access more than 65,000 vertices in a single draw pass, you have to split the draw calls. And unfortunately, since our surfaces are closer to 150,000 vertices per hemisphere, that means we have to do a lot of legwork just to be able to plot one of these surfaces. Another major limitation of WebGL, at least for this particular visualization, is that it does not have volume textures. This means we actually have to roll our own. We have to take our 2D texture call functions and actually be able to do this strange transform that lets us split it out into a mosaic of images and actually look up their locations through a function. So this unfortunately makes things much more difficult for us. But the saving grace for WebGL is that you actually do have access to a lot of additional OpenGL functionality through OpenGL extensions. So it wasn't until very recently did we actually get access to float32 render buffers and multiple render targets. And I'm planning to implement some really cool visualizations using these additional technologies. Another big downside of using WebGL is JavaScript's efficiency. So JavaScript is, in the end, it's an interpreted language, which means that it will be slower than C. If you were to take this exact same visualization, implement it in C, in Unity, it's going to be faster. But the good thing is, since we're already using all these kind of vertex and index buffers, JavaScript doesn't actually have to handle very much of it, except for just the loading side. So when you're loading the data, JavaScript has to kind of chew on it for a little while. But afterwards, it's silky smooth and super fast. So we don't actually directly use WebGL to do all of our draw calls. That's because WebGL basically just gives you a very thin wrapper to the GL calls directly. Kind of a pain to use when you're talking about a very large app like this. So we can actually use a library called 3JS that gives you very nice object-oriented wrappers for WebGL. And there's a lot of cool demos that have been implemented in 3JS. So if you just go to the website, 3js.org, you can actually see a bunch of them. So we have our nice framework that we can render all of our data with. And now we have to get data to it. Getting data into WebGL is tricky. That's because we have to make sure it is fast and it's small. because we're transferring everything over an internet link, we have to make sure that everything is highly compressed, but can be quickly uncompressed on the other side, because no one wants to wait 10 minutes while the browser downloads a 500 megabyte file to look at this one visualization. So we use a couple of tricks that both compress our data very small and also lets us open them up really, really fast. So when we're transferring our functional data, we actually do a mosaicing operation. So we take all of our slices of the data. It's a 3D volume. We actually take each slice and splay it out onto 2D in a certain configuration. And then we save these images as PNGs. This is great because PNG basically gives us for free zlib compression. That means that it's sparse. So all this area that's black out here, it takes up almost no space. Unfortunately, it's sparse in only the 2D direction. So even though there's sparsity through the plane here, it's not going to capture that very well. But it's good enough. And specifically, PNG gives us native support by the browser. Browsers were meant to handle PNG, so it can uncompress these images, dump them into memory buffers really fast, and there's no overhead associated. There's a small amount of overhead in the shader to actually allow us to look up the 2D location from a 3D coordinate, but we can deal with that. As for surfaces, we use a format called OpenCTM. This particular format lets us compress surfaces into a very small package. Specifically, it uses LZMA, which is a pretty state-of-the-art compression format, to really compress down the vertex data. It shuffles the vertices, it truncates them. It does a lot of tricks to get the majority of our vertex data down to only about 2 megabytes. And specifically, it also has a very nicely integrated JavaScript library, so I don't have to deal with trying to implement LZMA compression, decompression by myself in JavaScript, which would be very hard. So here's just a very simple metacode example of how I implemented the pixel mapper shader. So this is kind of the key thing. The vertex shader looks very similar to just a very standard vertex shader. We're going to pass all of our 3D locations through a frustum project and send them to our fragment shader. And this is where this thing takes over. So up here, you have a uniform texture object, the voxels data. So that's a texture object. Color map is basically how you're going to map from intensity to a color on the screen. And then you have that affine transform that translates from the surface to your functional data space. And then this is the vertex position that you get back from the vertex shader. So that's a varying inputs. That's what the vertex shader just sends you by default. You transform your vertex position into the functional position. You take it and you pass it through this mosaic lookup, which converts the 3D coordinate location into the 2D texture location. You look up the data in your texture, and you look up what color it should be, and send it to the screen. The actual code is a little more complicated than this, but this is really the core functionality. And you can see it's actually very simple. And specifically, it's so simple and easy that most graphics cards have no problem with it, even the laptop ones. So it's very fast to render these images. JavaScript does also give us a lot of advantages in terms of dealing with code that looks like that. So the code that I was using before was GLSL, which is the OpenGL shader language. And that shader language, unfortunately, doesn't really have much of a templating engine. So when we want to repeat code a whole bunch of times, especially in JavaScript, you can't really do that unless you access the code directly in JavaScript. So what we can do is basically metaprogram using the JavaScript to stack together pieces of the code in JavaScript and then send them to the graphics card to compile and render. JavaScript, unfortunately, is not great for templating. It's very slow when dealing with strings. But it's workable for this case since we only have to do it once every time you change how you want to look at the data. And specifically, I actually use it to handle how you deal with different data types, whether it's like a raw data format or something that you have to color map. And I use it extensively for thickness sampling. So when you have to take multiple random samples through the surface, I actually use JavaScript to template that so I don't have to write the code over and over and over. Finally, JavaScript gives you access to a lot of these interactions. So I don't know if you noticed, but I was using a leap motion to control the demo earlier. That means that since leap motion gives you JavaScript libraries, you can buy this little box for 70 bucks, and now you can have a touch-free interface to your visualization. And since everything is handled through JavaScript, and there's so many libraries out there for JavaScript, you can actually handle a lot of interaction just directly through JavaScript. So let me show you a demo that Alex set up actually a while ago.
If you go to the OGL demo,
yeah.
you go back.
So here's an interactive demo. This is using an older version of pyCortex, which is why you don't see the voxels, the square blocky things in the pixel mapper. But this is really nice because, like, unlike most visualization software, you can actually click on things and actually see what's going on, and also click on the other side to get other data. So what you're seeing here on the left, let me go back to the original data. What you're seeing on the left here is a map of what we think the brain is encoding in any specific spot, plotted into a special color map. So basically, things that are green in the brain here are things that we think the brain uses when it's thinking about people. Okay? And things that are purple are things that are in use when you think or see vehicles, for example. So this mapping actually was derived from an experiment that Alex did a while ago, but you can actually explore this data now. So let's say we want to see, what does this particular patch of the brain do? This is red, so we think it'll do something to do with people or places or something like that. And you can click on this particular voxel, and you can actually see that it does mostly do encode things about the outdoors and places and, you know, all sorts of different things. And you can actually explore the data in this kind of new unprecedented way, which is something that most software don't let you do. So for the future of pyCortex, now that we're actually getting access to a lot of interesting new OpenGL extensions, I'm planning to do some cooler demos using different shaders. So in this case, I've actually rendered a special shader. I've used a special shader that allows you to do some rudimentary volume integration through the cortex. So now, instead of having that thickness sampling where you just average together the samplings, we can actually see the individual voxels as they go through the cortical sheet. This was a really tricky render because it's actually using a lot of replicated shaders to try to get the thickness sampling and gets volume integration, but I'm hoping to have good access to this pretty soon. So look forward to more developments in pyCortex very soon. And now Alex will tell you a little bit more about how we actually do this research now that we have a way to look at it.
If anybody has questions for James, you should ask him now while I'm getting set up.
One of the things that looked like, like computational geometry, we do things called Delany triangulation, which actually separate things out, and then you can possibly go around your neighbors because you can sort of identify them and then do some sort of averaging or whatever. Does any of that make sense? Or is that
Yeah, so the sampling that we're usually doing is actually usually in the gridded space. So it's actually very easy to find the neighbors because they're in a grid.
Gotcha.
The expense comes from the fact that you have to do a lot of texture lookups, which is memory intensive and not something that shaders do well with.
Right.
So you can do, we have some early code that does implement some of that, but it's so expensive and it slows down the render so much that it's not great.
I didn't know if that filtering, if you were rotating, if you took that shape and you rotated it, it seems like the filter would give you different results. Is it rotation invariant? I mean, right?
Yeah, so trilinear filters are not rotation.
Right. So you sort of compute those one time and then stick with it, or
No. So the trilinear interpolation is actually computed by OpenGL. So it just uses the GL linear. It does bilinear filtering in plane, and then we do an additional linear step through the plane. So it's a very simple filter. It's unfortunately not radially symmetric, so we get corners, but it's such a small artifact that it's very hard to tell. Yeah.
Did you try doing, maybe mapping the images involved for two different people, viewing the same thing and check the similarity?
Yes, so actually that is something that one of our lab mates is working on right now, using canonical correlation analysis to take functional data between two subjects and try to co-register them. There's a lot of, in like MRI, there's actually a lot of efforts to try to figure out what are the similarities between brains. And the way that most people do it is not quite right. They actually usually take the volume space images and just kind of average them with the closest affine transform. And that's terrible because brains are not the same. But this technique that was developed by a lab mate Natalia Belenko uses basically partial least squares to try to match them as closely as possible, and that works pretty well.
Just a clarification, perhaps you answered this. The question is the following. So the demo that you showed us was just for one human?
Yes.
Okay.
Alex?
All right, thanks, James. All right, good to see you all today. I want to thank you guys so much for inviting us out here, Brent, and all the folks in the CS department. You've been awesome, and this is a really fun opportunity to go on a trip to South Carolina. And our friend Thomas Nazalaris back here, who worked in our lab until 2011-ish, yeah, who I think got us the hook up here and got us to come out. So thank you, Thomas, for doing that. So. James told you all about pyCortex, which was this visualization software that we're using. I'm going to tell you a little bit about some scientific work that I've been doing, which makes heavy use of pyCortex, and you'll see a lot of those kinds of visualizations through this. But really, the key thing that I'm interested here is language. Is like how is the meaning of language represented in the brain? So how is the semantic content? How is the meaning of the words that you hear being represented in your brain as you hear them. So I'm going to tell you about an experiment that I've been working on to look at that and show you some pretty cool results. All right, so this is the big question, is how is semantic content represented in the brain? Now, this is something that people have been talking about and sort of a question that people have been looking at for a long time. And there's a few things that we know really well about this. So this is actually a figure from a meta-analysis of, I think, around 300 fMRI studies that have been published on this topic. And this is a small fraction of the fMRI studies that have been published on this topic. This was only ones that met a certain set of criteria. But what they did here was, I guess you're all familiar with these pretty looking brains now, although they didn't use pyCortex here, for shame, is they plotted the centers of activations that people have seen for some kind of semantic task. So for most of these, it's things like, listen to a set of words, and in one condition you might judge whether the words have similar meaning. In another condition you might judge if the words start with the same sound, and then you contrast those two conditions and see which brain areas are more active when people are making semantic judgments about the meaning. So this is showing all the spots in the brain that people have found to be activated by these kinds of semantic tasks, and there are a lot of them. This covers like a huge amount of brain, it turns out, is involved in language. So in particular, we have here, this is the lateral parietal cortex, which is a really interesting sort of mysterious area. The lateral temporal cortex, and really just like all of the prefrontal cortex, which is super interesting. And the medial parietal cortex right here. So sorry, this is the left hemisphere, sort of this is looking from the side, this is looking from the middle, and this is the right hemisphere. In the right hemisphere, you can see that it's a similar kind of pattern, it just tends to be weaker in these experiments than in the left hemisphere. And that's kind of interesting, I'll touch on that a little bit later. So what we want to know is really what's going on here. So we know that this is representing something about the meaning of speech, but what is actually being represented there? What are those parts of the brain encoding about language, about speech, about meaning? So that's what I'm going to try to look at in this experiment. So, oh wait, sorry. One more thing that we know about semantic representations. So it seems like that there are a lot of parts of the brain that represent specific semantic domains. So this is a sort of weird term that I've come across kind of recently, and I really like it because it captures something that I think is meaningful about this idea of semantics. Is if a semantic domain is like a group of related topics or a group of related concepts, then it seems like there are some brain areas that represent semantic domains. So that there are places that represent sort of animals. So if you show people a bunch of different pictures of animals or a bunch of different animal words, you'll get consistent responses in some parts of the brain. Same with tools, but those are sort of the domains that we understand best, whereas we don't really know what other domains are, and people haven't really explored this space very systematically. In fact, a lot of what we know about this just comes from sort of lesion research, where people have brain damage in some part of their brain, and then suddenly they don't know anything about animals anymore, and then somebody said, oh, hey, that part of the brain probably does something that has to do with animals. So the hypothesis that I'm going to be working with here is that this domain selectivity, this sort of that parts of the brain respond specifically to certain semantic domains or encode specific semantic domains is widespread. That all of this semantic system that I showed you is actually doing something that's domain selective, but people just haven't tested the right domains yet because we don't really know what domains are. We don't really know, what constitutes a domain. So this is the hypothesis that I'm going to be testing here. Now let me tell you about the experiment. So in the Gallant lab, we like to do natural stimulus experiments. So this is where you don't show people really controlled things. Instead, you come on. Sorry. Let's turn that off. Instead, you just show people something natural, right? So when Thomas was in the lab, he was doing experiments where they showed people natural images. So you just flash natural images on the screen and people look at them, and then you extract some features from them and you build models of how the brain is responding to these natural images. In this experiment, we were using natural narrative speech. They're actually stories. So we just have subjects lay in the scanner and listen to a story for a few hours, actually. We have like 2-3 hours of data. These stories are actually from, if any of you are familiar with the Moth Radio Hour, it's an NPR show. It's also like a storytelling theater. So they have people tell true autobiographical stories, so stories about their lives, and then they record them and broadcast them on the radio. And we took the stories from the audience favorites collection from the Moth. So they're really good stories. They're really interesting stories. They're really and they're very well told. So it's really, this is the most fun fMRI experiment I've been a subject in, hands down, hands down. The ones that Thomas did were awful. They were just the worst experiments and just terrible. But this was really enjoyable. You just lay down, close your eyes, and listen to stories for a couple hours. So that was pretty cool. While the subjects are listening to stories, we record their it's called bold responses. This is blood oxygen level dependent responses, like James was describing earlier, using fMRI. This is the voxel size we used. This is what James described earlier as well. And we get an image about every 2 seconds. All right, so before we go further, I want to show you what this data looks like. Just what is the data, right? So this is a pyCortex viewer, you might realize. And over here, I'm going to show actually some snippets from some stories that we showed a subject. This subject happens to be James. This is James's brain, if you want to read something into that. So first, I'm just going to rotate the brain around so you can see sort of what this 3D structure looks like. You've already seen a lot of this. Flatten it out so we can see the whole thing.
We're bringing our children into. He thought it sounded like the kind of statement that brings people closer together, pointing as it did to their common fate. But the sexy mom just glared at him and took the healthy living supplement too, without asking.
So what I want you to notice here is that so the red and blue here are showing where there's high activity and where there's low activity. So red is where there's high activity, white is average, baseline, and blue is low activity. So what you should see as the subject is listening to these stories is that some parts of the brain come on at certain points. They sort of turn up their activity and some parts turn off. So let's hear another. There's some temporal interpolation, just linear temporal interpolation, and we shifted it by, I think, 3 or 4 seconds here. So it's supposed to match up pretty well.
In charge of the party always go, and you walk out saying less than 5,000 people in bathroom floors or
long overcoats, ski parkas, leather jackets, high school and college kids, having champagne 60-year-olds, linking arms and singing.
Only the 2 front windows with white shades lowered were not somehow blanketed. Your eye was constantly drawn to where the material converged mid-ceiling, punctured by a dazzling pink spotlight that looked like it might have just vaporized a flamingo. See?
All right, so this looks really incomprehensible right now, right? It's like, I don't know what the heck is going on here. If you try to match up what's going on here with something in the stories, that's hard. It's really hard. It's basically impossible. I've tried this. I've like stared at this for a while without knowing, really what each part of the brain is encoding here. It's just hard to do this matching. So we're going to get an algorithm to do this matching for us. We're going to use some techniques from sort of data mining and sort of computer science to try to match up properties that we might extract from the stories to the activity in the brain. So that's what I'm going to tell you about. And then at the end, I'm going to show you this with sort of better visualization, and hopefully you'll be able to maybe understand a little better what the brain is doing. All right, so this is the general framework that we use to do this kind of modeling experiment that I'm going to tell you about. So we call this voxel-wise modeling. This is the name that we settled on finally. We take the natural stimuli, like I told you about. We show it to our subject. We record fMRI data. We take these stimuli, and we also use some hypothesized feature space to extract features from the data. So, for example, we might, have a feature that's every time you hear the word there's like a thing that goes off. That would be a feature, or whatever else. In this case, we're actually going to use something that corresponds kind of to semantic domains. So this represents sort of a hypothesis about something we think is represented in the brain in these stimuli. Right, so we take the stimuli, we extract these features, and then in this estimation stage, we build voxel-wise regression models. So for each voxel in the fMRI data, we build a regression model that predicts its activity at each time point as a function of these hypothesized features. It's just a linear combination of these hypothesized features. All right, so this is the first stage of voxel-wise modeling, is estimation. Now what we do next is we want to test it. We want to see if this voxel-wise model works. Regression is a nasty thing. We might have like thousands of parameters in this regression problem, so we want to know that we're not overfitting to the data here. So what we do in the second stage, which is prediction, we take some new natural stimuli that the model hadn't seen before. We show them to the subject, record fMRI data. We push these stimuli into our feature space, and then we use the voxel-wise regression models that we'd estimated earlier to predict what the fMRI data looks like. So we're predicting, what the responses look like in this case. So we're not using the fMRI data to generate the models here. We're using the models to predict the fMRI data. And then we can compare these predictions to the actual fMRI data to see how good our models are. This gives us like a real value for like how much of the response to this natural stimulus are we explaining, right? And if we get to 100%, then that's amazing. Then we actually understand how the brain is representing these things because we're using natural stimuli, right? We're using sort of stimuli that put the brain into some states that are natural. Anyway, okay, so this is the second stage, is prediction. Now, if we find that the voxelized models are good, if we find that they're actually good at predicting the responses, then we might want to know why. We might want to know what kinds of features are actually being represented in each voxel. So then we can go on to the third stage, which is interpretation, where we take these voxelized regression models and we do something to look at what is actually being represented in each voxel. We can see like which features are mapping to each voxel. All right.
You don't have discrete semantic domains that you're trying to predict.
So that leads very nicely into my next slide. So how do we know which semantic domains are in the stories, right? So one thing we could do would be to, say, lay out a set of semantic domains. Like these are 100 different things that people might talk about, right? And then we go in and tag, each second of the stories and say, which of the 100 things does this belong to? That is, that's laborious for one and subjective because there is no good like catalog of semantic domains. So what we're going to do instead is use some techniques drawn from computational linguistics, which has a long history of sort of looking at semantic domains very successfully using statistical methods. So it's based on this idea called the distributional hypothesis. You shall know a word by the company it keeps. So this is basically that words that mean similar things tend to occur together in text. If you look across a huge amount of text, then words that occur together more frequently than you would expect, just by chance, tend to have related meanings. They're related to the same domain. They're related to the same topic. So using this kind of idea, we can get at semantic domains without having to, parcel out 100 different things that we might talk about. Instead, we can get a sort of maybe high-dimensional, smooth representation of semantic domain space. So that's what I'm doing here.
Yeah, thanks.
OK, so this is just a little example. This is a piece of text from one of the stories. And if we see words like eyes, skull, and hair, these words actually occur kind of close together. They're all in one sentence here. And they're all related. They're all parts of your head, right? So this is just the same idea. OK, so the way that I actually do this, there are a lot of different methods for doing this in the computational linguistics literature. I settled on something that was a little ad hoc and of my own construction. It happened to work extremely well for predicting this data, and it actually ended up being weirdly similar to some very sort of modern techniques that have come out of Google, which made me feel really good. But mine is much hackier than theirs. Anyway, so the idea is really simple. We have a set of target words. There are about 1,000 words here. These are words that sort of span the space of things that we might talk about. I actually took these from, are any of you familiar with the Simple English Wikipedia? Or Basic English Wikipedia? So it's Wikipedia in English, but written with a vocabulary of only 1,000 words, only with these words. So it's sort of a pared down, very basic, that people who are just learning English maybe would have a much easier time reading this than they would have reading like normal Wikipedia, which has tons of jargon and all kinds of stuff in it. So this is 1,000 words that sort of cover the set of things that we might talk about. So what I did then is I took my full lexicon. So this is all the words in the stories plus a few thousand other words that are just commonly used. And I found the co-occurrence between each target word and each word in my lexicon across a big corpus of text. So I have about 2.5 billion words of text. This is like, I have all of Wikipedia. I have a few hundred books that went in there. I have hundreds of millions of words of like comments that I scraped from internet forums, all this stuff. Just put it in there and then look for how often does each target word co-occur with each word in the lexicon within a 15-word window. So just how often do they occur sort of close to each other. These target words are things like difficult, husband, potato, and remember. And what this basically does is it gives us a score for each word in our lexicon, like how similar is this word to husband, or how similar is this word to potato. So we get like a potato-ness score or a husband score. And then this gives us a feature representation of each word. So each word now becomes a sort of 985-dimensional vector in this space, where words that have sort of similar meanings or are related to the same domain tend to have very similar vectors, and words that have very different meanings or related to very different domains tend to have very different vectors. So this is a sort of commonly used idea in computational linguistics, and we just took this and applied it to this kind of data. All right, so there's one more step here. So we take each word in the stories, and we look up its 985-dimensional vector in this matrix. But the words occur at sort of random intervals. So this is showing the first 60 seconds of one of the stories. And each of the little black bars here is one word, or it's showing like when that word occurred. So you can see that there's a sort of dense collection of words right here, and then there's a pause right here, and so on. So we need to get these 985-dimensional vectors into a sort of temporal domain that matches our fMRI data. So I just resample all the features. This is showing the value for one specific feature. I resample each feature at the acquisition rate of the fMRI, which is here 2 seconds, using just a sort of sync filter. So this is showing in black, this is the values for the individual words, and in red, this is the curve that's my resampled curve, showing sort of what's the average semantic domain over each 2-second period. All right, so that's it. This is our hypothesized feature space. This is how we get from the stimuli to something that we think might be related to the fMRI data. And it captures this hypothesis that I had that that semantic domains in the stories might be represented in the brain. So if we can use these features to predict the brain activity, then that is a good sign that semantic domains are actually being represented in those parts of the brain. All right, so then we estimate these regression models. We just, our predicted response is some, it's a weighted linear sum across our feature vectors here, x. And we estimate these weights using ridge regression, which is a standard technique, really easy to use, easy to apply. All right, so then we do prediction. We take this new story that the subject, that the model hadn't seen before, and we predict the responses on this story using these models that we fit. What I'm showing here is the flat map for one subject, where I've colored each voxel according to the correlation between our predicted response for that voxel and the actual response for that voxel. So the voxels that are gray here are ones that we didn't predict very well at all, and the voxels that are yellow or white are voxels that we predicted very accurately what was happening in those voxels during the story. So what you can see is that the models that I fit here work really well in these parts of the brain that I showed you at the very beginning in the semantic system. So we have like the lateral parietal cortex, the prefrontal cortex, the lateral temporal cortex, and the medial parietal cortex. And it's actually pretty balanced across the left and right hemispheres here. We don't have a big, it doesn't seem like left hemisphere is doing more semantic processing, or it doesn't seem like our models work better for the left hemisphere than for the right hemisphere. It actually, I don't think there's a significant difference between the two in this subject or in any of the subjects. Okay, so now we get to the final stage here, the model interpretation. And this is the most interesting. Now we want to know which semantic domains are actually being represented in each voxel in each part of the brain. So we can take a voxel and we can predict how it would respond to a bunch of things. This is a really simple way to try to interpret one of these models. So this is showing one voxel from the sort of inferior prefrontal cortex of this subject. And here I've just shown sort of a word cloud, words that the model predicts that this voxel would have a low response to, and words that the model predicts the voxel would have a very high response to. So it seems like this voxel sort of, it represents something about maybe numbers and distances, and it really doesn't like, you know, politics or culture or anything like that. So this tells us something about what is being represented in this voxel, or it seems to. But this is hard. We can't look at one of these for every single voxel in the brain. There's like tens of thousands of them, about 50,000. Yeah?
Did I understand this correctly? Are you suggesting that you're seeing it different between left and right brain in terms of?
No, sorry. This is just this one voxel right here. And then I'm just plotting, you know, along this axis which words I predict this voxel would have a low response to, and which words I predict this voxel would have a high response to. Yeah, so it's just within its one particular voxel. And that's actually the problem, is that this visualization is, you know, it's hard to look at. We have to try to figure out what this voxel is doing. And that's kind of difficult because there's tens of thousands of them. So we can't do this for every single voxel. So we got to do something to compress this information to get sort of a condensed way to see what is going on in each voxel. So a method that has been pretty successful in the past for doing this for us and for others is let's just reduce the dimensionality of these voxelwise models. So they started off as 985 dimensional models. So we have one weight on each of those 985 features I showed you earlier. But we can reduce it down to just a few dimensions using sort of standard dimensionality reduction techniques. Here I use principal components analysis, which is maybe the simplest of all dimensionality reduction techniques. It's just finding sort of what are the axes in the voxel models that explain the most variance across voxels. So what are the axes that let us represent the data most compactly using a small number of dimensions. So what I've done here is I've reduced it down to 3 dimensions. And here I'm just trying to interpret what these 3 dimensions are using, well here I'm using a color space where each of the dimensions maps onto one of the primary colors here. So the first PC determines the redness of each of these words, the second PC determines the greenness, and the third PC determines the blueness. And then this is showing just a few words from our space and where they project along these PCs. So this helps us sort of understand what this PC means, or what sort of stuff is being represented by the brain. What are the important representations in the brain? So this is hard to see. There's a lot of words here. So what I've done is I've actually simplified this down into just 12 categories using k-means clustering on these words. And I've given each category a name. This is just based on me like looking at the categories and interpreting them, but I've also gone back and have had subjects rate these categories and done a lot of things to try to figure out what these categories actually mean. But I think this is a pretty good interpretation of them. So it seems like the first PC kind of distinguishes between categories that have to do with people. So we have this like social, emotional, communal, violent categories, versus categories that have to do with maybe more perceptual things. So we have like tactile, visual, location, temporal, numeric, sort of more quantitative categories versus more social categories. So this is the biggest distinction between voxels in the brain. This is the most important distinction, is between social and sort of perceptual. And then along this other axis here, if we sort of, this doesn't quite line up with the first PC, but if we look along the other axis, it's sort of, we get categories that are not really, not exactly perceptual or social, but sort of in between. So down here we have this sort of temporal category, so this is time words, professional, which was a lot of words related to work, which was a weird category to pop out. And then up here, it's actually not captured by one of these 12 categories, but they're like words that describe body parts up here, which are sort of both perceptual and social in a way. Yeah, so what I'm going to do now is I'm going to take each of the voxels and project it into this 3PC space, and then color it according to this color map. And then we can see sort of what kinds of stuff all the voxels are selected for at a glance. So let me show you that. So this is the same subject that we've been looking at, but now I've colored each voxel according to this, and I had the categories up here so you can see that. So voxels that are red here, for instance, or voxels that are pink really prefer these like social and emotional categories. Voxels that are green prefer these like numeric, visual, tactile, locational categories, and so on. So these areas that we saw were well predicted before, like the lateral parietal cortex, prefrontal cortex, ventral temporal, and medial parietal cortex. They all show up as these sort of, they're not uniform. They're tiled with these colorful blobs. So it seems like there's a lot of sub-areas in here that seem to be representing different kinds of things. And that's really exciting, because these are areas that we haven't really understood well before. This lateral temporal cortex, this is one of these mysterious sort of association regions in the brain. That it doesn't really do visual information, it doesn't do tactile information, it doesn't do auditory information, it does something with everything, and it brings it all together into some sort of conceptual understanding of the world. And it seems like there are actually a bunch of different patches in here that are representing different kinds of things. And maybe even more interestingly, these maps, I was only showing you everything from one subject before, but this is maps from 3 different subjects. This is my brain, this is James's brain, and this is a third person in the lab. They're actually pretty similar, these maps. There's a lot of features that are shared across these brains. So we can see, you can pick out a lot of little things. There's a little purple patch right there, appears in every brain. There's a sort of dark red patch right there, appears in every brain. There's this green sort of ring around here, etc. So I've done a lot of extra work to sort of interpret what these patches are, what kind of stuff they represent, but I didn't think I had time to talk about that today. So I'm going to leave you with this. This is what I promised I would get back to you. This is the video that I showed you at the beginning that had the one subject, James's responses to this story. But now, instead of just showing the responses as this sort of red to blue color map, what I've done is I've actually colored each voxel according to its color in the PC map that I showed you a second ago. And the color will just get more strong when the voxel is active, and it'll get less strong when the voxel is not active. I'm showing the categories up here again so that you can see them. Now when I play this, what looked like sort of a mishmash of stuff before is going to become much more obvious, I think. It's not totally obvious still, but it becomes much more obvious what's actually going on. For instance, you're going to see when somebody describes the colors of something, for instance, you'll see these green regions pop out because that's a sort of visual thing. When they describe a person, then you might see pink regions pop out, etc. So let's take a look at this.
What a crazy world we're bringing our children into. He thought it sounded like the kind of statement that brings people closer together, pointing as it did to their common fate. But the sexy mom just glared at him and took the healthy living supplement too, without asking.
He put Lily in charge of the party while I was gone, and then he walked downstairs. There must have been 5,000 people milling around, wrapped in furs or long overcoats, or ski parkas, and other leather jackets, high school and college kids, heavily champagne 60-year-olds, linking arms and singing.
Only the 2 front windows with white shades lowered were not somehow blanketed. Your eye was constantly drawn to where the material converged mid-ceiling, punctured by a dazzling pink spotlight that looked like it might have just vaporized a flamingo.
All right, so I hope that tells you that we can understand a little bit more now about how the brain is representing what's going on in this story than we did when we started. All right, so just to recap, we saw that pretty much the entire semantic system is domain selective. That was the hypothesis that we started with. These domain-specific models predict most of the semantic system really well. It predicts all these brain areas really well. So it seems like domain selectivity is at least important in these areas. It's certainly not like the end-all, be-all of what these areas are doing. That would be silly, but it seems like there is some domain selectivity there. This anatomical organization seems to be consistent across subjects, and I didn't show you other results on that, but that is a really interesting thing that I'm looking at. And these previously identified semantic regions that were just sort of big blobs in our understanding maybe, now we see that they seem to be tiled with many distinct areas that represent different kinds of categories. All right, so I need to thank a bunch of people who are involved in this because I don't want to make it sound like I did everything. The Gallant Lab is a big group of people who all work together on everything. It's like a really tight-knit lab, which is really cool. We share a lot of expertise and code and data among everyone. Jack is a fun guy to work with. James, of course, did all the pyCortex stuff, which has been just totally vital for visualizing and understanding this. Also, I did a lot of this work with, in close collaboration with Wendy DeHeer, who's a grad student at Berkeley with Frederick Tunison, who studies audition, whereas in the Gallant Lab, really, we did vision. The Gallant Lab only did vision before this experiment, so this was a big step away from that. So Frederick was really invaluable, and with Tom Griffiths, who helped out a lot with the sort of computational linguistics end of things. And thank you guys for coming and listening. This is a lot of fun. All right. Yeah.
So 2 questions. First part is, what's kind of the end goal of this research? What would be the application of it? Like, how would it be used when it maybe
application? God.
And the second part is it seems like you guys have taken the visual approach. Mm-hmm. Now you've got the speech approach, right? Is there anything that is planned in the future for more of like how people interpret music in the brain or things like that?
That's interesting. So that's actually an experiment that a postdoc in the lab is starting up right now, is looking at music. So she has a bunch of piano music that's going to be played for subjects, and then trying to build a bunch of different models of how the brain is interpreting the music and sort of which different features in the music are being represented in which different brain areas. So that's going to be really interesting. It's tricky because MRI this is a weird constraint. MRI is very loud. It's just like the machine is extremely, extremely loud. It's like over 100 decibels when you're in there. We can do a lot to eliminate that sound. We have these sort of earplug headphones that we use, et cetera. But that doesn't totally eliminate the noise. With language, it's kind of okay because you can, as long as you can understand what is being said, it doesn't really matter what other sort of crap is going on in the background. But with music, that might be a little trickier. So we're still trying to figure that out. As far as applications of this, that's interesting. So one sort of obvious interesting thing is if we can now take these bold signals that we've recorded using fMRI and do the same kind of thing that James showed you a few minutes ago and reconstruct what the stimuli were from the responses. So if we can reconstruct the speech that somebody was hearing from their brain responses, that would be really cool. That would be a huge thing. If we got that working, then we might want to go to the next step, which would be reconstructing internal speech. So if somebody is just thinking something out loud, can you reconstruct that? We've done some initial tests, and the really weird thing seems to be that reconstructing internal speech might be much easier than reconstructing perceived speech. We get much bigger, more robust responses to internal speech when somebody is sort of actively thinking words than when they're just hearing words. So that is sort of in contrast to what, so this is very related to something that Thomas has been working on with imagery, where the responses to actual images that you see seem to be much stronger than responses to images that you imagine, and the inverse seems to be true with language. And that is probably because we have a system for producing language in our brains built in, and we don't really have a system for producing images, unless you're an artist, I guess. But yeah.
Well, actually, the whole, my wife tells me it goes through one ear and out the other. So,
yeah, exactly.
That does make a lot of sense.
It does, yeah.
Interesting.
Yeah.
Very cool.
Thank you. Yeah.
In terms of handedness, was there any difference amongst your subjects?
Oh, that's interesting, yeah. So actually, I had 7 subjects. 5 of them, I got like a dexterality inventory on everybody. 5 of them were strongly right-handed. 2 of them were weakly right-handed, so more on the sort of in the ambidextrous part of the scale. We didn't see any differences among any of these subjects. We excluded left-handed subjects from the start of this because we thought they'd just be too weird. They would throw things off with that many. Sorry. But it was surprising to see that there was just no difference whatsoever. From what I've heard, even the vast majority of left-handed people still seem to have the same sort of language organization in the brain as right-handed people, that most people still have this left hemisphere dominance for language, even though they're left-handed. And it's only a minority of left-handed people that have the switched dominance. So I think even with left-handed people, unless we get pretty lucky, we'll see the same kind of thing that we've seen here. But, you know, things weren't really very different left to right hemisphere either. It was pretty symmetric. So that'll be an interesting thing to look at in the future when we get more subjects. Yeah.
Is there any evidence in this of the regions in the brains and all language dependent or independent?
Whether they're language, so do you mean like?
So if you took 2 passages, one in French and one in English.
Oh, okay, yeah.
The regions being with the regions being close.
Yeah, so that's interesting. We're actually starting an experiment right now in the lab to look at that directly, where we're having bilingual subjects listen to essentially the same, or I think read actually, essentially the same story in English and in Spanish, and then build models like this of that data, and then see what, if the models overlap or if they're different. My hunch is that they're going to be identical, because I think everything that's happening here is sort of, it's past the level of words. It's not dependent on what the word sounds like or anything like that. This is really looking at the meaning of the words, and I think as long as that's the same between the 2 languages, everything that we see here is going to be the same. So that's my hunch anyway. There's a sort of different twist on that question. Are these representations specific to language, or are they, if you see something, or if you touch something, or if you hear, a dog barking, does that activate the same kind of representations? And that's something we're looking at too. So actually, I can take a bunch of the subjects I had in this experiment. I've also run another experiment where they watch natural movies, so they just watch movies for hours, and then we can compare the representations between those 2. And I actually have a slide here. Sorry, I kind of took this in a different direction because I wanted to show this slide. But so this is showing just on this one subject. So I took the movie data, I took the story data, and then I built these models for each voxel. And then here I'm showing the correlation for each voxel between the movie semantic model and the story semantic model. So the red voxels here are ones where there's high correlation. So the voxels seem to be responding to the same categories in the movies and the stories. Blue is where there's actually negative correlation, where they're responding to different things, which is really bizarre and interesting, and we don't know what the heck is going on there. But it seems like there's this sort of rim around visual cortex where they're really doing the same thing, and then a few scattered bits in sort of higher areas. But this is something that we're exploring further, and I think that's going to be really interesting looking at this. So like, which of these representations are really sort of cross-modal, high level, just like what is your platonic ideal of a dog, or yeah.
Well, both languages and music are hierarchical, and they follow power law, in particular this law for language. In music, kind of deviates basically, which would go tone music and software and bias, like it's called this law and stuff like that. So what I'm thinking is, and correct me here if I'm wrong, but fMRI data show that there's lots of pink noise, which is equivalent in this data, which is equivalent to this law. And so my thinking is this. Instead of looking at words and thinking that they're all equivalent, they're all similar, looking at some type of power law analysis, Zipf analysis, and finding, for example, the word this or the is very, very common. We hear it a lot. Mm-hmm. So, whereas a word like anthropomorphological is going to be a very rare word. And so, I suspect, I don't know, I'm not an expert, but over time, words like the, then we probably, the neural networks will kind of smooth it out and spread it all over the place. Whereas the word like logically might have a stronger presence. So taking into account the frequency of occurrence of words into the analysis might actually give you some better correlation.
That's really interesting, and your prediction is like so spot on. It's exactly right. So if I do this kind of analysis, but just so in the construction of my feature space that I used, I did a lot of things to try to normalize out word frequency. So I normalized by word frequency both of the target words and of the words in the lexicon. But still, some words just seem different. There's like the words like the, and this, and a, and so on that are just, they occur everywhere. They co-occur with everything. Interestingly, if we apply this same principal components analysis to just the stories themselves projected into this feature space, what comes out as the first PC is those words versus everything else, right? That's like the biggest difference in this feature space is just the function words, the closed class words versus all the words that actually contain things. And that just gets squashed by the brain. It just totally, the brain does not care about those words at all in this analysis. You can say the, a, whatever, it just doesn't, basically it just filters those out, they're gone. Yeah. So that's exactly as you said. Yeah, so I haven't looked at word frequency and how that maps to representations. That would be really interesting to do. I think people have done some things related to that, but I'm not sure of what the results are, and that would be really interesting to look at here too. Yeah, thank you. Yeah, Stephanie?
When you're looking at that, have you studied and have you noticed a difference between voice inflection and different subjects listening to male voices in the stories versus female?
That's interesting. So of the 7 subjects, I have 5 are male and 2 are female. The 2 female subjects are, I think, much more similar. The maps are much more similar looking to each other than to the male subjects, which is maybe not that unexpected. It's like the brain anatomy is different enough that they just sort of look a little different. But we haven't looked at the responses to male versus female storytellers. The stories that we used here were mixed, so it was, I think, exactly half and half male storytellers and female storytellers. And we haven't looked at inflection at all. Inflection is really interesting. I actually think that a lot of the response that we see is related to inflection, but we don't have good models for that yet. So that's something that we're really interested in looking at is inflection and sort of cadence, these micro pauses that people put in to indicate that something is supposed to be funny or that something is supposed to be sad or supposed to make you angry or whatever. There's a lot of subtlety in how people say words and how people sort of lead sentences that I think we could get a lot out of if we had good models of, but nobody knows what the hell's going on there. So that's an interesting problem. Yeah? Sure.
So the inflections, maybe just even the loudness, right? So inflection could be just a transition. So there's people tend to emphasize words. Anyway, a lot of other variables, right? They can sort of creep in.
Yeah.
You control the environment very well in the sense that the stories were interesting and the assumption was that the listeners were engaged rather than daydreaming or something like that, which could throw in some extra stuff that may not be representative of paying attention. But I see where you focus on that and try to eliminate those. But yeah, loudness, right?
Yeah. So actually, that's one of the really cool things about doing this kind of natural stimulus experiment is we can extract a bunch of different kinds of features from the stories and then fit models with all of them. So what I didn't tell you about here is actually when I fit this model, I actually fit other models alongside it. And so I had 2 other models that I fit on top of this one. There's one that's, well, I had 2 that were just sort of nuisance parameters that were the number of words per TR per image per 2-second period and the number of phonemes per 2-second period. And then I had one model that was actually the power spectrum of the sound, just in sort of 200 frequency bands, how much power is in each frequency band. averaged over a 2-second period, and another one that was which phonemes were spoken. So, did they say a S? Did they say a T? Did they say an whatever. So I actually fit those alongside the semantic model so that they can absorb all the variance that's related to those things. It turns out they only predict really in one place, which is right here in sort of lower auditory cortex, where you might expect. It's a nice control to have. They predict here really well. They predict a little bit in Broca's area, which is a sort of well-known speech area that seems to respond to a sort of phoneme content of speech. But yeah, so we did sort of look at that, but we didn't find much of the sort of loudness in these more semantic areas. But I imagine that there's a lot of, most of the loudness variation that we see is not like semantic loudness, right? It's mostly just sort of, is there somebody talking or are they not talking or something like that. So it'd be interesting to do something like, we could look at how loud this word was spoken versus how loud it usually is. Would be an interesting thing to see. Like, did they emphasize this word or was it just spoken normally? Yeah, we haven't tried anything like that. That'd be really cool. Yeah.
A few details. One is your co-occurrence was, you said 15 words?
15-word window. Yeah.
15 words. Okay. So that you're kind of leaving over any syntactic relationships.
That was exactly the point, to sort of exclude syntax.
Okay. And then the second thing that I couldn't help but noticing in the cool movies at the end, that you were getting some illumination in visual cortex occasionally.
Yeah.
Kind of this pukey lime green color.
Yeah.
Is that somewhere near the origin of all the PCs? Or is
So it is, the interesting thing is that, let's see if I can pull this up. This is actually the worst subject for visual cortex, but let's look at this, because this is a little better. I know this would be interesting to you. So what you might be able to see here is that actually in V1, right here, it tends to be this sort of pukey green color, which is visual, essentially. It's like a weak preference for visual concepts. The moment you go above V1, if you go into V2, V3, V4, LO, whatever, it flips. It becomes a preference for abstract categories that are unrelated to vision. So it's actually like suppressed by visual concepts. And that's this purple color here. And that's when I showed the maps of the correlation between the visual models and the story models. These purple places are the ones that are inversely correlated. So this is actually suppressed. When you hear words describing things, it's suppressed for the things that it would be excited for in the if you see them visually, which is super weird. I don't know. I don't want to like read too much into this because I don't really know what it means. I think it might be related to like closed eye movements or something like that, because the subjects just have their eyes closed and we're not controlling eye movements, and that's something that definitely happens. Is people move their eyes like differently to different topics or when they hear about different things. Yeah, but I'm not really sure, and that's interesting.
This activation that I saw in the one subject in B1 when their eyes were closed and they're just listening. Mm-hmm. Does that seem spurious or very subject dependent?
It seems to be very subject dependent, but this pattern of like sort of green visual-esque stuff in V1 versus purple like abstract stuff in higher visual cortex is there in everybody, just to very different extents. So you can see across these three subjects, this one has very strong purple stuff, this one has pretty weak purple stuff, this one has somewhere in between. Yeah, so it's more variable than almost everything else in the maps, for sure.
One more thing is just a geometric thing. If you were to close that map, I mean, you're really getting a band that kind of, I mean, there's some contiguity, right? You have these really beautiful bands.
Yeah.
Go from parietal down the temporal lobe. You go up into the medial wall. It seems to connect. It seems to sort of leave over the motor areas and then continue into the prefrontal cortex. It's like motor, somatosensory and motor kind of cut in half.
Yeah. So let's take a look at it on here. So this is I can't hide it. Dang it, this is too old. Hang on one sec. Can we hide the... No, that's not gonna work. Okay, well, you can kind of see here that, oh, there we go. So there's, no. Yeah, all right. I can hold this with one hand and point with the other hand. So this is the like big colorful patch in the medial parietal cortex. That's really the precuneus. And it seems to be bounded pretty sharply by, this is the edge of visual cortex. So everything below here is visual cortex. And on the top edge, this is actually somatosensory cortex is the other edge. So it's really like between those two, we get these really strong responses. We also get strong, like good models down here, and this is again the edge of visual cortex. So visual cortex kind of runs along here. I think what I've seen of like visual cortex representations seem to be more contiguous along this band than these linguistic representations. These are more like a little more anterior, so they kind of stop right here and then pick up again a little further along. Whereas the visual cortex, the like visual category representations seem to be more contiguous along this band, which is interesting.
This ventral band and that medial band, they're connected in the lattice, right?
Yeah. So they go along this, this whole thing right here. So this is the medial patch, and then this is pretty much contiguous all the way down to here. So this whole thing is connected. The connection through right here, this is IPS, is a little more tenuous, and it looks strong in this subject, but this subject has the weirdest organization in that area. This subject, usually this visually defined IPS area actually connects through to TOS right here, and then we see weaker representation of the linguistic stuff here. So it seems like the organization of IPS is kind of weird, that there's these intercalated sort of patches of like somatosensory representation, maybe more semantic. Like this is actually selective for I think numbers most strongly right here. So there's sort of number representation and then visual representation, and they're all kind of patched on top of each other. And maybe the exact pattern that they form right here is pretty different for different subjects. But yeah. Yeah, and then all of this is actually pretty disconnected from the prefrontal stuff. So there's actually this sharp disconnect right here. And this is right along the motor strip and the somatosensory strip, just cut between the prefrontal area that represents all the stuff there and this sort of parieto-temporal stream, which is cool. So I wanted to show you guys this real quick since I got it up now. This is a viewer that I made a little while ago with pyCortex, so we can see this is the map that I showed you before. But now what I can do is I can click on a voxel, and it'll actually show us the words that the model predicts the voxel will respond most strongly to. I apologize that it doesn't quite line up nicely. So this voxel really likes the names of like family members and words related to like drama, social drama. That seems to be a really common combination. Maybe it kind of makes sense because these are dramatic stories that they heard, so that's something that definitely shows up a lot. But if we look at these green voxels out here, we'll see numbers and sort of measures. If we look at these purple voxels, this is weird. This is really consistent across subjects. There's this weird little purple patch right there that really likes these kinds of emotional topics, which is interesting. And also words related to hearing. It's another weird one that's in this patch. So we get this like, where is it? I don't know. Yeah. So this is fun. This is the kind of things that you can do with pyCortex.
Have you like taken subjects and maybe just said those words to them? Just said those words and then just seeing what happens?
So I haven't done that. Yeah. No, I've been meaning to do that for a long time, but I haven't done that yet. I'm still curious. Yeah. No, that would be an obvious experiment to do to follow on to this. Yeah. All right. Well, thank you guys. It was a lot of fun.